The role of growth hormones in plants. Growth hormones are chemical substances that are produced in small amounts by the plant to regulate growth and development. Plant growth hormones include auxins, also known as the indole acetic acid, gibberellins, cytokinins, abscisic acid, and ethylene, which is a gas. Each hormone plays a critical role in the growth and development of the plant. So we look at each one of these and the contribution that they make, starting with the auxins. Auxins are produced at the growing tips, that is at the root and the shoot tips. Some of the effects include they stimulate cell division. Secondly, they promote cell elongation, thus influencing tropic responses. Now, tropic responses refer to the turning growth movements that is seen in flowering plants. So, for example, in this particular case, this shoot turns towards the source of light. So this is a case of positive phototropism. On the other hand, in this other setup, the shoot grows away from gravity. So this is said to be a case of negative geotropism. So this turning or bending of the plant parts is what is known as tropism. And the bending comes about because of an equal cell elongation caused by an equal distribution of auxins. So in this lower part, we have a higher concentration of auxins that stimulate more elongation. And that's how the shoot bends away from gravity. Same case in the case of light. On this other side, further from the source of light, you have more auxins. The more the auxins, the more the cell elongation. So the shoot tends to bend towards the source of light. So auxins play a central role in bringing about cell elongation, and in the process can influence tropic responses. And of course, cell elongation also contributes to the increase in length of the plant and also increase in the circumference. Another critical role of the auxins is the effect on the development of the lateral buds. The presence of the apical bud inhibits the development of the lateral buds into side branches. That phenomenon is known as apical dominance. So at high concentration, auxins cause apical dominance by inhibiting growth and development of the lateral buds. This apical dominance also includes suppression of lateral root growth by the main root. It occurs since the apical bud contains a high concentration of auxins which diffuse downwards. So from these apical buds, we have auxins diffusing downwards to the lateral buds below. The high concentration that accumulate at the lateral buds inhibits growth. This inhibition enables the plant to gain height to reach light for photosynthesis. So there is an advantage in apical dominance. It encourages vertical growth rather than lateral, so the plant can grow taller to reach light for photosynthesis. However, if the apical bud is removed, more lateral buds sprout into side branches as shown here. This is because cutting off the shoot apex removes the source of extra auxins that diffuse downwards to inhibit lateral bud growth. So the moment the apical bud is removed, like for example during pruning, if you prune a plant, you remove the apical bud, and in so doing remove the source of the extra auxins that would normally inhibit growth of the lateral buds. So the lateral buds develop into side branches, so the plant tends to grow sideways or laterally. Now this fact is put into practical practice. It has its application in agriculture, in pruning of tea and coffee, to encourage sprouting of the side branches, because the more the side branches, the more the tea leaves and the more the coffee berries that would be produced. Now, the second plant hormone that encourages growth are the gibberellins or gibberellic acid. Just like the auxins, gibberellins are a group of closely related chemical substances that exert the same effect on plant growth and development. Gibberellins are produced by young shoot and developing seeds. Some of the effects include: they promote rapid cell division at the internodal regions, thereby bringing about an increase in stem length. Now in this illustration here, this is a normal plant that is just allowed to grow normally, and this one has been treated by addition of gibberellins. Now these plants are seen to have grown taller than normal. This experiment illustrates that gibberellins do stimulate cell division and elongation at the internodal regions, bringing about an increase in the length of the stem. Another effect of gibberellins is breaking of the seed dormancy, and they do this by activating hydrolytic enzymes in the seed. Thirdly, gibberellins stimulate fruit development even without fertilization, even without pollination. As long as there are gibberellins, then the ovary will be stimulated to develop into a fruit. And development of a fruit without fertilization, thus producing seedless fruits, is known as parthenocarpy. Parthenocarpy is the formation of seedless fruits. Another effect of gibberellins is that it promotes development of lateral buds into side branches. So where we have a high concentration of gibberellins, a plant will tend to grow sideways because of the development of the lateral branches from the lateral buds. But where a plant has a low concentration of gibberellins, then it tends to grow straight because of the apical dominance, whereby the apical bud will inhibit the development of the lateral buds into side branches. But when we have sufficient gibberellins, then the lateral buds are stimulated to grow into side branches. And lastly, gibberellins stimulates flowering in many plants. Another hormone is also a group of closely related chemical substances that are known as cytokinins. Cytokinins are produced at the root tips, And the effects are promotional. They promote various aspects of growth by interacting with auxins. These promotions include cell division, flowering, and through cell division, cytokinins help to break dormancy in seeds and buds. Cytokinins also stimulate the enlargement of leaves. Ethene is another plant hormone. It is a gas at room temperature. It is produced by most of plant organs. The effects include the following. The most important, the ripening of fruits. It stimulates the ripening of fruit. This picture here shows bananas at different stages of ripening. This ripening is brought about by the hormone ethene. So all fruits ripen as a result of the presence of the gas ethene. So by enclosing fruits in a container, we prevent ethene from escaping, and the more the fruits stay, the more the ethene that they produce, and this will accelerate the process of fruit ripening. Ethene also promotes leaf and fruit fall in many plants. There are some plants, however, in which ethene may induce flowering, like in pineapples. And then lastly, you have the hormone abscisic acid. This is the main growth inhibitor in plants. It inhibits growth. It is produced in the leaves, stems, fruits, and seeds. So it is the main growth inhibiting hormone in plants. It also causes dormancy in seeds and buds. So when present, it will prevent the seeds from germinating, and it will also prevent the buds from developing. There are some survival values in prevention of growth and development, especially during the dry season. It will be important that the plant ceases to grow because water, for example, is scarce. So this inhibition of growth is brought about by abscisic acid. A third role is that it causes abscission or fall of fruits, flowers, and leaves, hence the name abscisic acid. Its principal function is the falling off. It causes the falling of leaves, especially during the dry season, or in some countries towards winter. During autumn, leaves fall off. This is in a bid to conserve water. And lastly, at high concentration, abscisic acid causes closure of the stomata. This is also during the dry season or when there is scarcity of water. Many plants will increase their levels of abscisic acid and in the process bring about the closure of stomata in order to conserve water.